This morning we're going to look at muscle physiology. It's the third group of excitable tissues. We mentioned that the excitable tissues are the nervous system or nervous tissue, the nervous tissue, the sensory receptors, and the muscle tissue. Today we're going to look at the muscle tissue. So we're going to look at the muscle physiology. Now, the muscle, in addition to being an excitable tissue, is contractile in function. So when it's excited, it contracts. A muscle is supplied with blood vessels and also nervous and neural tissue. Now, muscle perform 5 main functions. They help maintain posture. As we sit or stand, we are being supported by the muscle, and the muscle is helping us to maintain posture. Secondly, they stabilize the joints. They stabilize the joints. Then they produce movement. Movement from one part to another, or movement in the body, are all regulated by muscle. Then they generate heat. As a result of the muscular activity, heat is generated, and this will help us to regulate body temperature. And finally, they guard entrances and exits. There are sphincters which will ensure that these entrances are guarded. And these sphincters are made up of muscle. Now we'll look at the characteristics of muscle. First, they are stretchable. You can stretch muscle, just like you can stretch any elastic material. Then they are elastic. You stretch muscle and they come back to their original length. So they are elastic. They are irritable, meaning that if you stimulate them, or they can be stimulated, they respond to stimulation. So they are irritable. Then finally, they develop tension. When muscle contracts, they develop tension, and this tension would aid us to do work. Now, classification of muscle. Muscle can be classified functionally or structurally. Functionally, there are 2 types of muscle. We have voluntary and involuntary muscle. Involuntary muscle cannot be regulated or by consciousness, right? They are automatic. They are automatic in function. Voluntary is when we consciously regulate the muscle. Now, based on this muscle, we have cardiac and smooth muscle, which are involuntary, and skeletal muscle, which is voluntary. Then structurally, muscle can be classified by the presence or absence of striation. So functionally, we have striated muscle and we have smooth muscle. Now, basically, there are 3 types of muscle. We have cardiac muscle, which are striated and involuntary, and are found in the walls of the heart. So the cardiac muscle are found in the walls of the heart. We have skeletal muscle, which are striated and voluntary. They are attached to the skeleton, and they help in movement and maintenance of posture. Then we have smooth muscle, which are not striated and involuntary. Now, these line body cavities. So basically, these are the three types of muscle we find in the body. Now we're going to look at skeletal muscle. We're going to look at skeletal muscle. I mentioned the skeletal muscle are voluntary and striated. Right, now we're going to look at the skeletal muscle. So it's a specialized tissue that has both ability to contract. No. The skeletal muscle are the muscle that attach to the skeleton. They are voluntary and striated. Now, if we take a muscle, the whole muscle is covered by a connective tissue called epimysium. So this is our muscle, our whole muscle, and it's surrounded or covered by a connective tissue called epimysium. Now, the muscle is made up of fascicles. So these are the fascicles in the muscle. And this is a singular fascicle. And the fascicle is covered by a connective tissue called perimysium. Here, we have a perimysium surrounding a fascicle. Then the fascicle is made up of nerve cells. It's made up of nerve cells it's made up of muscle cells. It's made up of muscle cells. And they are covered by a connective tissue. A collection of muscle cells are covered by a connective tissue called endomysium. So we have endomysium. Now, the muscle cell is made up of myofibrils or fibrils. So these are myofibrils. And that's a myofibril. The myofibril is then made up of myofilament. Okay, I'll go over it again. I mentioned that we are going to look at the skeletal muscle. The skeletal muscle is the muscle that attaches to the skeleton. It is striated and voluntary. Now, if we take a muscle, a whole muscle, it is covered by a connective tissue called epimysium. So this is our whole muscle, and this is our epimysium. Surrounding the connective tissue surrounding the whole muscle. Now the muscle is made up of fascicles. So as you can see, muscle physiology, please. We are doing muscle physiology. As you can see, the whole muscle is made up of fascicles. These are the fascicles. Right. And the fascicle, each fascicle is surrounded or covered by a connective tissue called a perimysium. So this is our fascicle, and it's surrounded by perimysium. Then the fascicle is made up of muscle cells. The fascicle is made up of muscle cells. And a collection of muscle cells are covered by an endomysium. The muscle cells are covered by a connective tissue called endomysium. Then the muscle cell is made up of fibrils or myofibrils. So these are the myofibrils. Then the myofibrils are made up of myofilaments. So these are the myofilaments. And there are two types of myofilaments. These are the thick filaments and the thin filaments. So now we're going to look at the muscle cell or muscle fiber, the skeletal muscle cell or muscle fiber. Typically, that's the skeletal muscle cell or muscle fiber. It is cylindrical and elongated. And it has a lot of nuclei, which are peripheral. They are not central, so that's a nucleus. Take a skeletal muscle, that's another nucleus. Okay, this is it. I said, from our previous lecture, we said that there are three types of cells which are excitable. We have the nervous cell, we have the sensory receptors, and we have the muscle cell. We've looked at the nervous cell, we've looked at the sensory receptors. Today we're going to look at the muscle cell. So muscle physiology. And I mentioned that the muscle, in addition to being excitable, is contractile in function. So you excite a muscle and the muscle will contract. And the muscle performs 5 major functions in the body. It helps us maintain posture. Sitting down, walking about, standing upright. These are all ensured by muscle so that we don't topple over. That's how it helps us maintain body posture. Then it stabilizes the joints. It produces movement. If we move about, the movement that occurs in the GIT, the contraction and the beating of the heart, and movement that occur in the blood vessels are all regulated by muscle. Then muscle generates heat as a result of the activity. In the muscle, heat can be generated, and this becomes very important during temperature regulation. And lastly, muscle guard entrances and exits of the body. Between the esophagus and the stomach, there is a sphincter. There are sphincters in almost every part of the body, and these are made up of muscle. So they help guard entrances and exits. Then I mentioned the characteristics of muscle. We said they are stretchable. You can stretch a muscle. Right? It's like it's elastic in character. Then if you stretch it, it can return to the original length. So that is how come we say it is elastic. It's not plastic. If you stretch plastic, it can't go back to its original length. But an elastic will go back to its original length. Then muscle is irritable, meaning muscle will respond to a stimulus. It responds to a stimulus. And muscle do develop tension. When they are stimulated, they develop tension. Now, classification. Muscle can be classified functionally or structurally. Now, functionally, muscle can be voluntary or involuntary. Then structurally, it can be striated or smooth, that is, without striation. Now, there are 3 types of muscle. These are skeletal muscle, which is striated and voluntary, and these attached to the skeleton. Then we have cardiac muscle, which is striated but involuntary, and they form the wall of the heart. Then we have smooth muscle, which is non-striated and non-voluntary. These line body cavities. The smooth muscle line body cavities. Now we're going to look at the individual muscle structures. First, we'll look at skeletal muscle. But before I start the skeletal muscle, did you understand what I just said? From the functions, characteristics, and classification. Is it all right with you? Good. Now, skeletal muscle. They are attached to skeleton and they are responsible for our movement and maintenance of posture. Now, if we take the whole muscle, which attaches to the skeleton via the tendon, the whole muscle is covered by a connective tissue called epimysium. So this is our skeleton and it has epimysium. Now, the whole skeleton is made up of fascicles. So these are the fascicles, and that is one fascicle. The fascicle is covered by a connective tissue called perimysium. Now, the fascicles are made up of muscle cells or muscle fibers. And this is an individual muscle fiber. Now, the muscle fibers, a collection of them, is covered by a connective tissue called endomysium. Now, the muscle fibers are made up of fibrils. So these are the myofibrils. A collection of them will be found in the muscle fiber. Now, the myofibrils are made up of myofilaments. Now, there are two types of myofilaments. These are the thin filaments and the thick filaments. So, structurally, that's how the skeletal muscle looks like. So, we have the muscle, which is made up of fascicles, and the fascicles are made up of muscle cells or fibers. The muscle fibers are made up of myofibrils, and the myofibrils are made up of myofilaments. Now, let's look at a typical skeletal muscle cell. The skeletal muscle cell is elongated, unbranched, and cylindrical. It has almost all the organelles that are found in the typical cell. The skeletal muscle cell is multinucleate. Means if you take one skeletal muscle cell, there are a lot of nuclei. And these nuclei are peripheral. They are not central. They are found well at the periphery, like these. Now, the central portion of the muscle fiber is occupied by the myofibrils. So these are the myofibrils, which have pushed the nucleus to the periphery, the nuclei to the periphery. The skeletal muscle fiber has a lot of mitochondria, you see, which are also at the periphery. Now, each myofibril is surrounded by a system of tubes called the sarcoplasmic reticulum. So these are sarcoplasmic reticulum. So this is sarcoplasmic reticulum. The sarcoplasmic reticulum has swelling ends. Those are the swelling ends. So these are the tubular system that surround the the myofibril, the swollen ends. The swollen ends of the sarcoplasmic reticulum are called terminal cisterns. Terminal cisterns. And the sarcoplasmic reticulum is the storehouse for calcium ions. Calcium is stored in the sarcoplasmic reticulum. Now, on the surface of the skeletal muscle fiber, there are invaginations or holes. Now, these invaginations get into the skeleton, and they are an extension of the plasma membrane of the skeletal muscle fiber. Now, these tubes that enter this cell are called T-tubules. They are called T-tubules. And it is via these tubules that action potentials will get from the surface of the cell into the cell. Now, each T-tubule is sandwiched between two terminal cisterns. And these three structures constitute what we call the triad. Now, going back to this diagram, this is our terminal cistern, this is another terminal cistern, and they are sandwiching the T-tubule. So these two terminal cisterns with the sandwiched T-tubule constitute what we call a triad. And the triads are found at the AI junction in the sarcomere. Now we'll get to know what the AI junction is. Now the whole muscle fiber is surrounded by a sarcolemma. So that's our sarcolemma. And in the sarcolemma, we have the T-tubules, invaginations that get into the cell. We have our myofibrils, which are surrounded by the sarcoplasmic reticulum. Then we have the T-tubules being sandwiched between two terminal cisterns to form the triad. The nuclei in the cell are pushed to the periphery of the cell. They are not central. Then we have also a lot of mitochondria found in the cell, which are also peripheral. Now, the sarcoplasmic reticulum are the storehouses for calcium ions. And action potential is conducted into the cell via the T-tubules. We're going to look at the myofibrils. We said that the myofibrils are found in the muscle fiber. The muscle fiber is mainly made up of the myofibrils. And the myofibrils are made up of two filaments, myofilaments, the thin and the thick filaments. Now, it's the arrangement of the thick and thin filaments that gives striations to the cell. So this is a myofibril from this muscle cell. You can see that there is an alternating dark and light bands. These alternating dark and light bands give the muscle the striation characteristic. So these are our striations. Now if we take a single myofibril, this is how it looks like. Now this appearance is as a result of the way the myofilaments are arranged in the myofibril. Now let's look at a simplified diagram of a typical myofibril and the arrangement of the myofilaments. So the myofibrils have thick and thin filaments. The thick filaments give what we call the A band. So these red are the thick filaments. So from the thick filaments are arranged to give us the A band, which is the anisotropic region of the skeletal muscle. So from here to there is the A band. Now, within the A band, there is a lighter shade region, which is just made up of only thick filaments. And this region within the A band, which is lighter colored, is the H zone. So we have our H zone in the A band. Our A band starts from here to there, and it's made up of thick filaments. And at the edges, we have an alternation between thick and thin filaments. So this region appears darker. Now, this region appears lighter, even though it's in the A band. And this lighter portion of the A band is the H zone. Now, within the H zone, there is a darker portion here within the H zone called the M line. The M line holds the adjacent thick filaments together. So we have our A band, we have our H zone, and within the H zone, we have the M line. Then we have the I band. The I band made up of thin filaments. Here. So I band continues. The thin filaments. Within the I band, there is a dark portion called the Z line. The Z line holds the adjacent light myofilaments together. So the thin filaments are held together by the Z line. Now the area between two Z lines, from this Z line to that Z line, constitutes what we call a sarcomere. A sarcomere, which is the functional unit of the muscle. So this sarcomere would stroke across the whole muscle fiber, giving rise to the striations. So you can see this. This is a thick filaments, this part. This is the thin filament, and this is the Z line. There is also the thin filaments, thick filaments, Z line. The region between two Z lines, as we've said, is the sarcomere. And within the A band, there's a lighter portion, which is the H zone. And within the H zone, there is a darker line called the M line. The M line holds adjacent thick filaments together. Then within the I band, which is made up of the thin filaments, there is a very dark line called the Z line. And the portion of the muscle from one Z line to the other constitutes the sarcomere, which is this functional unit of the skeletal muscle. Now, this arrangement of the myofilaments give rise to the striations found in muscle. Okay, I'll go over it again. If we look at our muscle, this is a muscle fiber. It's striated. You can see bands of lines along the skeletal muscle. Now, this striation is due to the arrangement of the myofilaments in the myofibril. So if we take out our myofibril, this is what it looks like. We have bands, and these bands are the striations. Now, we are going to look at a very simplified diagram of the striations. Right. And we have an A band. This is our A band. And the A band is made up of thick filaments. So these are the thick myofilaments. They constitute the A band. They constitute the A band, the thick myofilaments. Thick myofilaments constituting the A band, which stretches from here to there. So we have our A band. Now within the A band, in a relaxed muscle, there is a lighter shade, a lighter region within the A band. Now this lighter shaded region within the A band is called the H zone. So you can see that this region is lighter, and it's called the H zone. Now we have the H zone because at the edges of the A band, we have alternating thin filaments with thick filaments, this region. It's a region of overlap between thin and thick filaments. So in the region of the A band where there is no overlap between the thick and thin filaments, there's a lighter shaded region. And this lighter shaded region is called the H zone. So from here to there within the A band is called the H zone. Is it clear to this point? Is it clear to this point, please? Feedback. Is it clear to this point? Good. Now we have another band made up of just thin filaments. Now the region of the muscle made up of just thin filaments is called the I band. So we have the I band here because it's just thin filaments. I band here because it's thin filaments. Now the I band is made up mainly of thin filaments. Then within the I band, we have a dark portion. Within the I band, there is a dark portion. And this dark portion is called the Z line. Now the Z line holds adjacent thin filaments together. So you see the Z line holding the thin filaments. Now the region in the muscle from one Z line to the other is called the sarcomere. The sarcomere, which is the functional unit of the muscle. So from one Z line to the other Z line is the sarcomere, which is the functional unit of the muscle. Now, in the Z zone, the H zone, in the H zone, there is a thick line called the M line. Now, the M line is protein that holds the adjacent thick filaments together. So we have a M line. Now, this arrangement is found all over the muscle. Right. It's found all over the muscle. Now, every single thick filament is surrounded by 6 thin filaments. That's a cross section of the sarcomere. The thick filament, each thick filament is surrounded by 6 thin filaments. And each thin filament is surrounded by 3 thick filaments. So basically, this is the way the myofilaments are arranged in the cell, in the myofibrils, which gives us the striations in muscle. Now that we've seen the myofibril, we saw the muscle fiber itself, we've seen the myofibril. And we said the myofibril is made up of myofilament. Now let's look at the structure of the myofilament. I mentioned that there are 2 types of myofilaments: thick myofilaments and thin myofilaments. Now let's look at the thick myofilaments. The thick myofilament is made up of 2 heads and a tail. Now the myofilaments are held together, and this gives us the thick filament. Now the thick filament is made up of the tails of the myosin, protein myosin. The thick filament is made up of the protein myosin, which has a tail and 2 heads. Now all the tails will come together to constitute the thick filament. And sticking out of the tail, we have the heads. Now the heads have 2 sides, main sides. There is an actin binding site and an ATPase site. Now the ATPase site is an enzyme. It hydrolyzes ATP. And the actin binding site will bind to the thick filament, which is made up of actin. So in summary, the thick filament is made up of the protein myosin. And each protein myosin has a tail and 2 heads. Now the myosin proteins come together to constitute the thick filament. And the thick filament is made up of the tails of the myosin proteins. And sticking out of the tail, we have the heads, 2 heads. And on each head, there is an actin binding site and ATP binding site. Is that clear to this point? The thick filament. Is it clear to this point, please? Feedback. Good. Now we're going to look at the thin filament. The thin filament is made up of 3 proteins. These proteins are actin, tropomyosin, and troponin. Thin filament is made up of 3 proteins: actin, tropomyosin, and troponin. Now we are going to look at the individual proteins. The actin filament, which usually constitutes the thin filament, is made up of spherical proteins. And these spherical proteins get together to form a chain, just like the rosary. You know, the rosary is made up of beads stringed together. So the actin molecules are spherical proteins which come together to form a long chain. Now the thin filament in the skeletal muscle or muscle is made up of two of these chains coming together. Two of the actin chains coming together, and these two chains form a double helix like this. So we have two long actin chains coming together to form a double helix. The actin filaments forming a double helix. Now, the tropomyosin filament is a long protein filament, and it lies in the groove of the actin chain. So we have the tropomyosin lying in the groove of the actin chain. Now, in the resting muscle, the tropomyosin covers the myosin binding sites on actin. Now, the actin filament has a myosin binding site. So you see binding site for the attachment of myosin. And on myosin's head, there's an actin binding site. So the tropomyosin chain will cover the myosin binding site of the thin filament or actin filament or actin chains. Good. So that we've seen the first and the second proteins. Now the third type of protein found in the thin filament is troponin. Troponin has three subunits. It has troponin T, troponin I, and troponin C. Now the troponin occurs at regular intervals along the thin filament. Now troponin T binds to tropomyosin. So troponin T will bind to tropomyosin to form the troponin-tropomyosin complex. Troponin I binds to the actin filament, and this holds the tropomyosin in place to cover the myosin binding site. And troponin C will bind to calcium ions. We got to know what the significance of troponin C when you're doing muscle contraction. Now, is it clear to this point? We've seen the actin double helix, we've seen the tropomyosin, now we've seen troponin C. Good. So this is the arrangement of the proteins in the thin filament. We have a double helix chain of actin, and we said the actin is made up of spherical proteins. Now intertwined with the double helix actin filament, which forms the thin filament, is the tropomyosin chain. Now the tropomyosin chain covers the myosin binding site on actin. Then we have the third protein, which is the troponin. Now troponin has three subunits. The troponin complex has three subunits, which are troponin T, which binds to the tropomyosin filament to form a troponin-tropomyosin complex. Then we have troponin I, which binds to the actin filament, thus holding the tropomyosin in place. Then we have troponin C, which will bind to calcium ions. So this is our thin filament. Now we've seen the arrangement of the thin and thick filaments in the skeletal muscle. Next, we're going to look at the contraction of the skeletal muscle. But before that, I want to know if you've gotten all that I've said from the first slide to the arrangement of the proteins so far. Now we're going to look at the contraction of the skeletal muscle. Now, three main events occur in the skeletal muscle that would result in contraction. First, they have to be excited, electrically excited. We saw excitability in the nervous system when we're doing action potentials. Secondly, we have the excitation-contraction coupling, where the excitation would initiate contraction. Then we have the contraction proper, where tension will be generated in the muscle. First, let's look at the excitation, the electrical excitation of the skeletal muscle. Now, the resting membrane potential of skeletal muscle is negative 90 millivolts, and I guess you know what the resting membrane potential is, since I spoke about this at length when we're doing the nervous tissue. The resting membrane potential is negative 90 millivolts, right? And when this is stimulated, it gets depolarized. Now, the action potential of skeletal muscle is just like in the nervous system. When it's stimulated, there's opening of voltage-gated sodium channels, there's influx of sodium ions, and we get reversal of polarity. Then we have repolarization. And I don't need to talk about this since I talked about it last week. Now, when the muscle is stimulated by a motor neuron, usually through transmitters, without the depolarization, and an action potential will be initiated. And you know the action potential. The action potential is initiated in the sarcolemma, that is the plasma membrane that surrounds the muscle fiber. Now, when the action potential has been initiated, it triggers the next phase, which is the excitation-contraction coupling. The action potential will be conducted into the muscle from the sarcolemma via the T tubules. And I guess you know what the T tubules are. So, action potential is generated on the plasma membrane, and it's conducted into the cell via T tubules. So these are T tubules. Action potential is being transmitted across into the cell. Now, within the T tubules, there are some voltage sensors. Now, these voltage sensors are calcium channels, and they are called dihydropyridine receptors. So this is a dihydropyridine receptor, which are voltage sensors in our calcium channels. When the action potential gets to the dihydropyridine receptors, they are activated. Now, these dihydropyridine receptors, when they are activated, will activate ryanodine receptors in the sarcoplasmic reticulum via electron dense feet. It means, what this means is that the dihydropyridine receptors are coupled to the ryanodine receptors in the sarcoplasmic reticulum by electron-dense feet. So when the dihydropyridine receptors in the T tubules are activated by the action potential, they also activate ryanodine receptors in the sarcoplasmic reticulum via the electron-dense feet. Ryanodine receptors will open and calcium ions will flow out of the sarcoplasmic reticulum into the sarcoplasm. Now the calcium, when it gets into the sarcoplasm, will initiate the contraction proper. We've seen the excitation. Now the excitation contraction coupling, which is the how excitation would initiate contraction. So now we have calcium being released into the sarcoplasm. Now the calcium that is released into the sarcoplasm will bind to troponin C. We said troponin has 3 subunits: the troponin T, the troponin I, and the troponin C. So calcium binds to troponin C on the thin filament. When calcium binds to the troponin C on the thin filament, the troponin undergoes a conformational change. And this change will shift the tropomyosin filament on the actin filament. When the tropomyosin filament shifts, the myosin binding sites on the actin filament are exposed. When these are exposed, they can interact with the myosin or thick filament. I'll go over that again. Calcium is released from the sarcoplasmic reticulum. Calcium binds to troponin C. When calcium binds to troponin C, the troponin molecule undergoes a conformational change. Now, this will sort of break the link between troponin and actin. And the myosin filament will shift. After troponin undergoes the conformational change, the myosin filament shifts from its original position. When the tropomyosin filament shifts from its original position, the myosin binding sites, which were covered by the tropomyosin filament, are exposed. Now, the next stage of contraction. We saw that the myosin head has an actin binding site and an ATP binding site. So ATP, now we're going to look at the cross-bridge cycling. And the cross-bridge cycling, the cross-bridge is the head of the myosin. The head of the myosin is called the cross-bridge. And the cross-bridge has a myosin binding, actin binding site and an ATP binding site. So ATP binds to the cross-bridge, and because the ATP binding site is an enzyme, it will hydrolyze the ATP into ADP and inorganic phosphate. Now this reaction energizes the head of the myosin and would increase the affinity of the myosin head for actin. So the breakdown of ATP into ADP and AP, inorganic P, energizes it and this will make the myosin head bind to the myosin binding site on the actin filament here. So the myosin head will bind to the exposed myosin binding site on the actin filament via its actin binding site. Now when the myosin binding site, the myosin head binds to the actin, the inorganic phosphate is released, and this triggers what we call the power stroke. On the power stroke, the head of the myosin would rotate and pull the thin filament across the thick filament. And during this process of power stroke, the ADP is released. Now, even though the ADP is released, the myosin head still attaches to the actin filament. Now, the process where the inorganic phosphate and ADP is released is called the product release. Now, next, ATP will bind to the myosin head. When ATP binds to the myosin head, the affinity of the head for actin decreases. So the head will detach from the actin filament. Then the ATP will be hydrolyzed to ADP in an organic phosphate, and the process will continue. Because the process, as long as ATP is present in the skeletal muscle, this process will continue. And this process is what we call the cross-bridge cycling. And the turning of the head and the power stroke will make the thin filament glide over the thick filament. And this results in the generation of tension during contraction. Now I'll go over it again in a complete cycle. Originally, the myosin head has an actin binding site and an ATP binding site. So when the myosin binding sites on actin have been exposed, the next stage is the cross-bridge cycling. ATP binds to the myosin head. To be specific, it binds to the ATP site, which is an enzyme. Now, this ATP site would hydrolyze the ATP into ADP and inorganic phosphate. Now, when this is done, the myosin head is energized, and it develops a high affinity for actin filament. So the myosin head with the ADP and inorganic phosphate will bind to the actin filament here. When the myosin head binds to the actin filament, the inorganic phosphate is released. So we have only ADP now attached to the myosin head. Now, this brings about what we call the power stroke, where the myosin head will turn about 90 degrees. And as it turns, it drags the thin filament across the thick filament. So our power stroke will draw the thin filament across the thick filament. And the ADP will be released from the myosin head. Next, ATP will bind to the myosin head again. And when ATP binds to the myosin head, its affinity for the thin filament decreases. So the myosin head will detach from the actin filament. when it detaches from the actin filament, it will hydrolyze the ATP to get ADP and inorganic phosphate, and then bind to the thin filament again. So the cycle is repeated as long as ATP is present in the cell. Is it clear now? So now we've seen the contraction of the muscle. So this is our power stroke. It binds to the thin filament, and the tenon will drag the thin filament across the thick filament. And this will result in the decrease of the width of the sarcomere. Now, the development of tension and shortening of the muscle can be explained using the sliding filament theory. Now, in the sliding filament theory, would have the thin filament being pulled over the thick filament. So this, assuming this is a relaxed muscle, that's a sarcomere. These are Z line, our thin filaments, and our thick filaments. So we have our actin and the myosin. Now, the myosin head will bind to the actin filament. Then we have our power stroke. The power stroke will drag the thin filament over the thick filament. So the movement will be from here to the next point. Then the myosin head detaches. It is activated again, binds to the thin filament, and draws the thin filament in, closer to the M line. So if we began with a relaxed muscle like this, this is a partial contraction. You realize that the H zone is decreasing in width. Till at a point when the muscle is fully contracted, there is no H zone. So we start from a sarcomere of this width to that of this width. This is the sarcomere, that's the sarcomere, and this is the sarcomere when the muscle is fully contracted. So it's like those of you who've lived close to the river or sea, if you have seen the dugout canoe, or even if you watch the Olympics, the boat and the paddling of the boat, assuming the boat is the thick filament, and the river or water is the thin filament. The paddle, when you dip it into the water and drag at it, gives you the power stroke that will draw the canoe in the opposite direction. And as a result of the detachment binding power stroke, the thin filament will slide over the thick filament, and the sarcomere, which is the functional unit, will decrease in width as a result of this cycling. So if the sarcomere of a relaxed muscle is about this width, during muscle contraction, the sarcomere will decrease in width. Okay, it will decrease in width, and that is the sliding filament theory.